So, my research assignment by Kate Quiroga. So, my major is on biological sciences, microbiology. My area of interest is on vaccine development, infectious diseases, and pathogenesis. The specific department at UMD is the Department of Microbiology and Immunology. So, the College of Computer, Mathematical, and Natural Sciences. So the professor that I looked into is Dr. Yuxing Li. She's an associate professor at UMD, and her research field is on B-cell response to viral infections, HIV pathogenesis, and antiviral therapy. So the title of one of the research that she has done is on abnormal B cells. So the title was Abnormal B-cell memories, memory subsets dominate HIV-specific responses in infected individuals. So what I got from this is that abnormal B-cells subcategories are main contenders to the HIV-specific responses in these infected individuals that differ from those that are not infected from HIV. So what I also learned is that it's hard to make an anti-HIV antibody that are memory cells. And also little is known about HIV antibodies that will neutralize the HIV. So for the abstract, so an experiment was done. So they used GP140, so that's just an HIV with no without a transmembrane. So this is glycoprotein, and this glycoprotein would bind to a B cell and B cells receptors. So there's a co-receptor, so COR, and there's a CD4 receptor on the B cell. So there's an overactivation of the memory B cell that will produce antibodies, because B cells produce antibodies, but the emergence of these two receptors are different. So this one comes about much earlier, but it's a poor neutralizer, because it makes antibodies that are poorly neutralize the HIV. And then there's CD4 receptors that come later in HIV that are good at neutralizing, because it makes good antibodies that neutralize. So yeah, they basically bind to different receptors. So these bind to different receptors, and that will lead to some differing neutralizing. So the production of making antibodies, so HIV antibodies. So here we have the HIV antigen being engulfed by the phagocyte, right? Through endocytosis, and this is going to become, the phagocyte becomes an antigen presenting cell, which an active, a helper T cell will bind and connect and then become active. So this will go and lead to a humoral immunity response, which will go to some B cell, and that B cell will therefore make memory B cells and plasma cells. Those plasma cells will make antibodies. So these are the HIV antibodies that will bind to some antigen, HIV antigen, and neutralize it, either being a poor neutralizer or a good one. So some background knowledge to know about HIV. So HIV stands for Human Immunodeficiency Virus, which causes AIDS. So the detection of HIV can be done through HIV testing. So it will detect HIV antigens and HIV antibodies. So these are produced by your immune system when you're first exposed to some bodily fluid of an infected individual, either by one of these 5 things. So sexual contact, or inheritance, or injection of a drug, or others. So HIV spreads throughout the body through viral RNA integration into T cells. So the HIV has RNA in it. And when a T receptor binds to a HIV receptor, it's going to give its RNA, viral RNA, to the T cell through retrotranscription. So it's going to turn that RNA to DNA, back to DNA. So the opposite of transcription or protein synthesis. And this will lead to the viral DNA being included or integrated into the T cell, and that then the T cell will replicate or make more copies of the HIV cell and make many of those, and this will continue to go on. This will also worsen the patient, leading into chronic HIV. So now that we know more about HIV, we could look at the research that was done. So let's look at the research axis. So for the research question, or the conceptual question precisely, do abnormal memory B cells lead to specific responses in HIV-infected individuals? There's also, do the abnormal antibodies' inability to neutralize the HIV contribute to the HIV-infected response? And finally, are the receptors' shape conformation a contributing factor to the HIV-specific responses, whether the antigen HIV receptor that binds to the B cell receptor, causing the abnormalities of the antibody. For the experimental question, how do abnormal memory B cells lead to specific response in HIV-infected individuals? For the hypothesis of the experiment is, if the abnormal memory B cell leads to specific responses in HIV-infected individuals. For the second hypothesis we have, if abnormal antibodies' inability to neutralize the HIV contribute to the HIV-infected responses. And for the last one we have, if the receptor's shape conformation is a contributing factor to the HIV-specific responses. Now, looking at the method in the experiment, they use flow cytometry. So they use an antibody for a specific marker, so an IgG antibody, and that will attach to a specific antigen, HIV, and this will detect these things: size, shape, count, and other properties. So how was the method taken? So a sample was taken of HIV-infected individuals or non-infected individuals. So in group 1, there was early infected HIV vermic, but with no ART. And then for group 2, we had chronic HIV vermic plus no ART. And then for group 3, we had early infected plus those who started ART. And group 4, chronic HIV infected plus ART. So ART is a way to alleviate the HIV virus or prevent it from becoming AIDS. So there's 2 methods of checking for HIV. So there's DNA assay, and there's a phenotypic analysis and cell sorting. So this one was through centrifugation, and DNA assay was assessing the B cells' response to influenza or tetanus vaccination. So, for the different methods of HIV detection, there's phenotypic analysis and cell sorting. So this, as I said, was through density gradient centrifugation. So they performed using an MABS. So this would identify the GP140, so that's the HIV with no transmembrane, a mutation of CD4, so that was on the B cell, and that would just change the shape of the receptor, and then a single mutation on this co-receptor that was on the B cell as well. So it would be colored, and it would use flow cytometry analysis. So for the low density assay, there was transcriptional analysis of the B cell and HIV cell. So it looked at 29 genes, chosen genes that would probably be contributors to HIV. So RNA was isolated from the B cell and HIV B cell. So this would determine the hierarchy clustering and heat map visualization of memory B cells that which were performed using heat maps. So here's a schematic of the method. So this is flow cytometry. So there's atoms going through a tube, and a laser would shoot to the cell, and this would give a picture, and it would detect the size, shape, and count or properties of the cell. So it would say like when the IgG antibody binded to the infected B cell by the HIV. So for transcriptional analysis, there was DNA that was extracted from a community, and they got specifically one portion of the DNA that contributed to the HIV virus, and through PCR it amplified the fragments, so it made many copies of that HIV specific gene, and then through that it was used in an experiment in 2 different conditions, and then they would extract the RNA and reverse transcribe it, so that retrotranscription, and then they'd try to find what parts were transcribed, and yes, that's it. So the prediction is then receptors of CD4 receptors are not developed, or they're developed when the infection is worse. So they develop late, and for receptors of co-receptors on the B cell they're developed early, but those antibodies produced by the B cells are non-neutralizing to the HIV. So the claim is the first one is abnormal memory B cells lead to specific responses in HIV individuals. The second, abnormal antibodies are unable to neutralize the HIV, which contribute to the HIV-infected responses. The third one, the receptor shape conformation is a contributing factor to HIV response. And the fourth one is the abnormal antibodies are produced from infected memory B cells that are unable to neutralize the HIV. So for the evidence, we have 2 groups, the infected, HIV-infected, and non-infected, and 2 groups, the GP140s without a transmembrane that bind to the B cell. So we see the antibody detection of them by attaching to a cell and exchanging foreign material. In division B, we have GP140 and a point mutation on the HIV receptor, CD4 or the co-receptor. So there's 2 groups, vermic and non-vermic. So during early stages of HIV, antibodies are produced and bind to receptors of the HIV that neutralize it. And antibodies are still produced at a relatively high number. During late stages of HIV, antibodies are not frequently produced, and antibodies produced aren't effective. They decrease on the number of antibodies produced.